Hi there, so my name is Thiv and I'm a medical student and I've done a previous degree in medical genetics. So can everyone see the screen?
Yeah.
Yep, great. Okay, so today I'm going to be talking about the role of genetics and what it does and what it tells us about COVID-19. So what we're going to look at is what is genetics, what does genome sequencing mean, what do we know about the disease, what's a mutation, who's at a higher risk, and why certain people are getting it more than once, and how genetics plays a role in vaccines. So a gene is basically a segment of a DNA that codes for a specific protein. So different genes lead to different proteins. And genetics is the studying of these genes and the pattern of inheritance. And a genome is the full sequence of DNA that's present in an individual. So genome sequencing is often done to analyze the different DNA sequences. So it can tell you a lot about the human sequence, bacteria, viruses, and identify the similarities and differences using a database. And it tells you how different diseases are inherited and so on. So genome sequencing, why is it useful? So as I said earlier, you can, if a person, let's say person A becomes ill with a viral disease, you collect the viral sample and you sequence the genome and you use a reference and see whether it already exists in the database. If it does, then you've got all the information you need in regards to that virus. If it doesn't, then it can tell you how similar it is to another virus or any other microorganisms that it's got a reference point to, and tell you which exact sequence is different, and maybe that's the disease. So what do we know about COVID? Caused by, stands for severe acute respiratory syndrome, a virus called coronavirus, and it was first discovered in December 2019 in Wuhan, and it is an RNA virus. So an RNA virus is a single-stranded to led to a global outbreak between 2002 and 2004, and COVID-19 has got us, the virus has got a 96.2% similarity to the bat coronavirus, which some people may think that that could be where the disease came from, but researchers believe that this may have mutated and had another intermediate host and then have been infected into humans afterwards. So it didn't directly come from bats. So what else do we know? We know that it's got the spikes on the virus is what binds to the human's enzyme called angiotensin converting enzyme 2, which is usually found in the lungs and some other organs. And the viral load, which is the amount of virus particles present, is usually found in the upper respiratory tract. And we also know that it's got an R0 value of 2.63. So the R0 value tells you how many people are likely to be infected from one infected person. So if there's one person that's already infected, they can infect around about 3 people. And this was the number initially in May 2020, but now it's believed to be 0.9 to 1.2, and the number can change according to how the population behaves to the virus. So if you go through lockdown, then this value is going to decrease as you're less likely to spread the virus. So SARS and versus SARS-CoV-2. So we know that both viruses have got these spike proteins, and this is what binds to the host. And what are the differences in terms of the disease itself is that the SARS in 2002 to 2004, the viral load was detected in the 2nd week of the illness, which means that you became ill and then you are more likely to spread the virus to someone else. Whereas with COVID-19, you're more likely to spread the virus before you even know that you have the symptoms. Therefore, it's harder to stop the spread of the virus. And if we look at the numbers, I believe this one was about total cases was about 8,000, I think, whereas this one is already in the millions. So the genetics, so we know that there is a similarity, but the difference in that sequence could be what increases the viral load and the severity of the disease. So moving on to mutation. Mutation is basically the mistakes made by the virus when it replicates its genetic information. And mutation isn't necessarily a bad thing or a good thing. So what happens is you have these sequence here. So you have ACG, for example, and that produces a specific protein. And when you replicate this, if there is a mistake and it becomes AAG, then it's possible that it could make the same protein, a different protein, or no proteins at all. So knowing that the viruses can mutate doesn't really necessarily tell us whether it can cause a more severe disease. But what we know is that with CoV-2, SARS-CoV-2, the mutation rates are lower, which is a good thing for us. So it can help us with vaccines. And even if it does have fewer mutation rates, it's even more unlikely for these new strains, new types of viruses to be any more advantageous. So the different variants, so research was done, and what was found is that initially at the beginning, there was a specific strain of the virus, which was D614. All that means is that at position 614, there was this amino acid here, and that was the predominant variant at the beginning of the pandemic. And as the time goes on, the G614 became more predominant. And what does that mean? Well, one thing we know is that as you can see on the diagram here, it has a higher viral load, which means that if you have G614, you're more likely to spread it than D614 to someone else. But it doesn't tell us, there is no significant difference between the severity of the disease. Am I at a higher risk if a family member has COVID-19? From a genetics point of view, the risk is the same in terms of there isn't a specific gene in your family that you inherit which can predispose you to the disease. However, if you live in the same household as the family member, then that will increase your risk as you are in the same sort of environment. So you need to make sure to ventilate the rooms, keep them open, avoid contact, wash your hands, and follow the regulations. Is the Black, Asian, minority ethnic group at a higher risk of COVID-19? And the reason why this is a very important question is, as we know, a lot of the cases and the fatality rates are mainly seen in this group. So again, research has been done using genome sequencing to see if there is any sort of specific sequence or genes involved that could possibly lead to a more severe form of the disease or lead to fatality. And so far, there isn't any genes that are associated with this. So what does that mean from a genetics point of view? No specific group is at a higher risk, but if that's the case, then why is it that the Black, Asian, minority group is more at risk? That's because of their environment mainly. So that's to do with the socioeconomic status. So poorer socioeconomic status is shown to have poorer health outcomes. It also depends on where you live. So if you live in a densely populated area, then you're more likely to spread it to other people because the population is much higher, and also the line of work that you do. So key workers are more exposed to the disease. So therefore they're more likely to get it. And as we can see here, the diagram shows, so the red is showing the population of BME in London. And as you can see in the food production section, 54% are of that group, and key public services and transport workers, a lot of them are from this community. So they're more likely to be exposed. This brings on to why are people likely to get COVID more than once? So as we know, the first time you get it, you're likely to build antibodies to this. And we don't know yet how long these antibodies last in our system. So the reason could be, one, these antibodies could decrease over time, and the second time you get infected, you don't have the antibodies to fight it off. Or it could be that you have a different strain of the virus. So as we looked at before, the first time it could have had D614, and the second time it could be that you have G614 strain that infects you. And the third theory is that the antibodies produced from the first time could essentially lead to a pathway for another infection, but that's a theory. So there's no cause or anything being found for that yet. Genetics and vaccines. So what do we know, and what goes inside these vaccines is a question that everyone's asking at the moment. So in the Pfizer and BioNTech vaccine, there's something called a messenger RNA or mRNA. So what that is it's a segment of the DNA that codes for the virus spike only, so just the spike proteins in the virus. And what it is once you've been injected, these spike proteins are made, and then your antibodies learn to fight these off as it recognizes them as foreign. And the adenovirus is basically a vector. So why are we using a vector? It's because it's more like it's got a higher chance of producing an immune response. So therefore the adenovirus is used, and it's the same principle where you put the genetic material in to make the spike proteins, and then your body produces the antibodies necessary for to fight that off. So then when you are infected with the actual virus, you've already got the antibodies necessary to fight it off and less likely to produce the symptoms. And yeah, that's the end of my presentation, so thank you. And if you have any questions, feel free to ask.